So, all right, I think our apologies for these like technical issues, but I think Naomi can just like start over.
Okay, so I can continue now. Hopefully it will be okay. Okay, so I was talking about the second thing that I like to consider. I think we should consider when studying lipid metabolism, which is the fact that membranes are plastic, right? And this means that cells adapt their metabolism and are able to rearrange their lipid composition very efficiently upon different stimuli. We can, of course, follow these lipidome rearrangements using lipidomic analysis, and we can translate this into what I'm showing you here in this volcano plot, where we can see, and I will be using this kind of plots through my presentation, where we can see the lipids that are being downregulated upon certain stimuli, and at the same time, we can actually see how other lipids are upregulated. And so these lipidome rearrangements are actually so efficient, I believe, because now we know that indeed all the lipid metabolic branches or the different lipid metabolic branches are actually interconnected to each other, which in practical terms means that if we perturb a particular lipid synthetic pathway, the cell is going to adapt and create a response to this perturbation. And I believe, and I hope I can convince you today, that indeed following this adaptation of the cell to these perturbations can give us a lot of information. And we can unravel, on the one hand, new networks of co-regulated lipids, and on the other hand, we can potentially even uncover new lipid functions. And so this project started with a very simple question, which was, what happens to the cells when we deplete the sphingolipids? Or in other words, which are the cellular responses to sphingolipid depletion? In order to tackle this, we use a highly interdisciplinary and collaborative approach. We use, on the one hand, our targeted lipidomic analysis approach in the lab, where we can measure around 600 lipids in each run, and we can measure many different glycerophospholipid species, as well as many sphingolipids, as I will show you. We combine this approach with a high-throughput genetic screen in collaboration with the lab of Jonathan Weissman. And in his lab, Manuel Leonetti, who was a postdoc at the time, was as well interested in understanding the cellular responses to sphingolipid depletion, and he performed a genome-wide CRISPR screening of cells treated with miriocin, which is a very specific inhibitor of sphingolipid metabolism, and I will come back to this in a minute. I will be showing you again the data set coming from the screening in this kind of volcano plots, where we will be able to see in the left those genes whose knockdown induces hypersensitivity to miriocin treatment or sphingolipid depletion, and in the right we will be able to find those genes whose knockdown induces resistance to sphingolipid depletion. And finally, I will be showing you, among other techniques, also molecular dynamic simulations coming from the lab of my collaborator Stefano Vanni here in Switzerland in the University of Freiburg. So, okay, why am I so interested on this particular lipid class, sphingolipids, and what are these sphingolipids? So I'm showing you here the structure of a ceramide molecule, which is a sphingolipid, of course, as opposed to the structure of a more commonly found, let's say, glycerophospholipid, phosphatidylcholine. Sphingolipids are composed by a sphingosine-based backbone here that can be acylated through an amide bond to form ceramide. And we can further modify this molecule here in this hydroxyl group to attach different head groups that will give rise to sphingomyelin and other more complex glycosphingolipids. The reason why I'm so interested on these lipids is because first, they have very particular physicochemical properties that makes them have a very complex membrane behavior. I don't have time to go into this, but I'm showing you an example of a giant liposome here, in which we can see the phase separation that ceramide induces when we add this lipid into these liposomes. These sphingolipids are also bioactive. This is the sphingolipid pathway, and now we know that many members of this pathway, like sphingosine-1-phosphate, sphingosine, or ceramide 1-phosphate, are what we call bioactive lipids, which means that they are able to very efficiently activate or inhibit signaling cascades that are going to be crucial for the fate of the cell, such as proliferation or apoptosis. This regulation of sphingolipid levels has been found in many pathological conditions, and moreover, mutations on the enzymes of these pathways are known to cause very severe genetic diseases. So the importance of these lipids for cell physiology is pretty clear. As I told you before, in order to deplete the sphingolipids from our cells, we use this inhibitor, myriosin, which is a very specific inhibitor of the first enzyme of the de novo sphingolipid synthesis, the serine palmitoyl transferase. So, okay, let's take a look to what happens when we deplete the sphingolipids, which are the cellular responses. This is gene ontology enrichment from the data set coming from the screening. And I will be focusing today mainly on this side of the plot because we already could see very interesting things from the beginning. First, we find many enzymes or proteins involved in cholesterol biosynthesis. And this for us was a great proof of concept because the functional and genetic interaction of sphingolipids and sterol metabolism was already shown years ago by Howard, actually. Second, we were very interested on this other group of hits that were essentially proteins involved in the metabolism of other lipids, and I'm going to come back to this as well in a minute. And finally, this other group of proteins called as well our attention, which are proteins involved in different steps of intracellular trafficking. And I will as well, of course, come back to this. I don't have time to go in deep into discussing all these hits, but I like to show this plot because I think it represents very well the robustness of the method, since we find enzymes involved in the same pathway being either hypersensitive or resistant, depending on the substrate specificity, in particular in the acyl chain specificity of the substrate, the chain length specificity. We see this for the elongases, for example, those enzymes that elongate fatty acids. We find cells lacking LOBL5 being hypersensitive, while cells lacking LOBL6 to be resistant. We see a similar phenotype for acyl transferases. AGPAT1 is on one side and AGPAT2 is on the other side. And we find a similar thing for phosphinosidide metabolism. We see that cells lacking PI4 kinases are hypersensitive to sphingolipid depletion, while cells lacking the corresponding phosphatase SAG1 are actually resistant. But what happens actually with the lipidome of the cell when we deplete the sphingolipids? What we can see here is that indeed myriosin is really efficient depleting sphingolipids. We see all the sphingolipids we can measure decreased, and we see mainly two lipid classes being upregulated when this happens at the same time. I'm going to be focusing today on one of these lipid classes, which are ether lipids. And this is the data in mole percent now, so that you can also have a taste of the relative amounts of these lipids. And we see that indeed, when we deplete the sphingolipids, we have a very consistent increase on ether PC. In order to see how consistent this is across different cell lines, we did the same treatment in different cell lines, and we see that indeed this response is pretty consistent and universal. Moreover, when I do the lipidomics of these, some of these resistant hits here in the right side of the plot, we see that these cell lines have increased baseline levels of ether lipids, suggesting actually that having a lot of ether lipids can make cells resistant to sphingolipid depletion. Finally, but very important, we think the lipidomics data correlates very well with the genetic data because we find in the screening that cells lacking this enzyme, AGPS, are actually hypersensitive to sphingolipid depletion. And AGPS is indeed one of the first enzymes involved in ether lipid synthesis in the peroxisomes, suggesting indeed a synthetic lethal effect between the two metabolic branches. Okay, so what are actually ether lipids, which are the main focus on of the talks today? Ether lipids are a subclass of glycerophospholipids that are characterized by having an ether bond that can be ether or vinyl ether, as opposed to the more commonly found ester bond in canonical, let's say, glycerophospholipids. This difference arises by the fact that for their synthesis, the cells use a fatty alcohol instead of an acyl-CoA. These lipids are actually very abundant in mammalian tissues. They can range from 5 to 20%. And interestingly, they are also intermittently distributed across the evolutionary tree. So this is something we actually don't understand very well. More interesting for us was the fact that these lipids are present in membranes not only as free lipid components, but they can be attached to proteins. And this is the case of glycosylphosphatidylinositol anchor proteins, or GPI anchor proteins. These proteins are soluble proteins that are synthesized in the endoplasmic reticulum, and they need to be secreted following the secretory pathway up to the plasma membrane or the extracellular media. In order to do this, and since they have to follow the vesicular transport, they need to be attached to membranes, and they do it in mammalian cells using a lipid anchor that is indeed an ether lipid. So as I told you, these proteins are attached to membranes and they need to follow vesicular transport. And the first step is the COP2-mediated vesicular transport from the endoplasmic reticulum to the Golgi apparatus. These proteins cannot bind directly the COP2 machinery, and so they need an adapter or a receptor. And the adapter for the export of GPI anchor proteins is the P24 complex, which in mammalian cells is comprised by these TMETs, being TMET2 and TMET10, the most important subunits for the oligomerization of the complex. And so in the membrane of the endoplasmic reticulum, the P24 complex recruits and binds GPI anchor proteins, packages them into COP2 vesicles, and brings them to the Golgi. Now, once in the Golgi, the P24 complex needs to be recycled back to the endoplasmic reticulum. And work from the lab of Britta Brugger and Felix Wieland some years ago described how, in order for this recycling to be efficient, the P24 complex needed to bind a very particular sphingomyelin, which is a sphingomyelin with 18 carbons at the SN2 position. And so this is a good time to introduce you the last group of hits that called our attention from the miriosine screen, which were proteins involved in different steps of intracellular transport. Of course, we were interested on the ones involved in the early secretory pathway, which is this transport from the ER to the Golgi. And I hope you can appreciate that we even find the P24 subunits here, confirming actually a role of sphingolipids in the homeostasis of the P24 cycle. So the next thing we asked was, okay, we see an upregulation of ether lipids when we deplete sphingolipids, but is this happening the other way around? So for that, Manuel made this CRISPR cell line for the AGPS, this enzyme that we find in the screening, and we confirmed by lipidomic analysis that indeed this cell line has decreased levels of ether PC or ether lipids. And interestingly, we find that indeed ceramides are also upregulated in this cell line. We see an increase in ceramides, and even though the major sphingomyelin species in these cells is not significantly affected, we see a very particular decrease in a specific sphingomyelin, which is this sphingomyelin with 18 carbons at the SN2 position that I told you, it's the cofactor P24 needs for an efficient retrograde transport back to the endoplasmic reticulum. And so, okay, sphingolipids and ether lipids seem to be metabolically linked and co-regulated. And we hypothesize at this point, integrating all the data I just showed you, that maybe this is happening because these lipids are actually working together to regulate a particular pathway in cells. And we hypothesize that this common function could be modulating the secretion of GPI anchor proteins. In order to test this hypothesis, we took advantage of the RASH system of retention using selective hooks, which is essentially a way of synchronizing the export of your protein of interest. So we use as a reporter a GPI anchor protein that is hooked to the endoplasmic reticulum through a streptavidin binding system. And then when we add biotin to our cells, our cargo gets released and we can follow the kinetics of the secretion of our protein of interest. And we can measure the arrival to the acceptor compartment, which in our case is the Golgi apparatus, which is labeled with a resident protein, which is GM130. And so this is how the experiment looks like in a wild-type situation. And I hope you can appreciate that after 10 minutes of biotin addition, we start seeing our construct arriving to the Golgi. We can measure thousands of cells because we use a high-content microscope from the Access facility here in the department. And this is the quantification for a wild type. And the first thing we did, kind of as a proof of concept, was to knock down P24 and see what happens. And indeed, what we found was that there is no export of GPI anchor proteins, even after 45 minutes confirming the role of P24 as a carbon receptor for these kind of proteins. So now what happens when we deplete the sphingolipids or we deplete ether lipids? So what we see is an acceleration of the secretion of these proteins. They leave the air faster, in a faster way. And this effect is cumulative because when we do both treatments at the same time, the effect is bigger. So suggesting indeed that these lipids might be working in the same pathway, but perhaps actually in different steps of the same pathway. And so we hypothesized that indeed what could be happening is that the P24 complex might be binding sphingomyelin 18 in order to have an efficient retrograde transport to the endoplasmic reticulum or recycling. And maybe what is happening is that the same P24 complex is binding ether lipids for an efficient anterograde transport of GPI anchor proteins. In order to test this, we teamed up this time with computational biophysicists in the University of Freiburg here in Switzerland, and Stefano Vanni and Valeria Soni, a fantastic PhD student in his lab, tested this hypothesis. First, they checked if actually the transmembrane domain of P24 was binding sphingomyelin 18 under their conditions. And once they confirmed this, they built our system, which I'm showing you here is composed of POPC as a membrane scaffold. We have ether PC here in black and the transmembrane domain of P24 in blue. I'm showing you the data in these density plots, density maps, and you can see our lipid of interest, ether PC, in blue in those regions where the lipid is depleted, and in red in those regions where the lipid is enriched. And this is the quantification of the depletion enrichment factor, just take into account that if this factor is higher than 1, this means that the lipid is enriched, and if this factor is lower than 1, the lipid is depleted or not enriched. And what we could see is that if we take all the membrane into account, ether lipid is enriched, but ether PC is enriched, but it's not very significant. And maybe you can already appreciate here that when we take into account only the luminal leaflet of the membrane, indeed this enrichment gets really significant. And this for us was great, and this is really important because indeed we know that the P24 complex binds and recruits GPI anchor proteins in the luminal leaflet of the endoplasmic reticulum. So I've shown you so far how using actually high throughput genetics combined with an exhaustive lipidomic analysis, we were able to metabolically link to structurally very different lipids, sphingolipids and ether lipids. We believe that the cells are co-regulating these lipid species because they collaborate or they work together regulating a certain process, which we believe is the secretion of GPI anchor proteins through the binding to P24 complex. And I told you that GPI anchor proteins in mammalian cells are actually ether lipid based, which makes a lot of sense. However, what I didn't tell you is that this is not the case in every organism. And indeed in yeast, we know that the lipid anchor of GPI anchor protein is a ceramide molecule, which is a sphingolipid. So we were shocked when we realized that indeed the co-regulation we find in mammalian cells is in a way conserved through evolution, where a ceramide molecule has been substituted by an ether lipid to perform the exact same function as a lipid anchor of GPI anchor proteins. And I believe that nature is actually choosing this difference in these different lipids in this particular case, probably because they share common physicochemical properties or effects on membranes. And in order to test this, we went in vitro in reconstituted membrane system using this Flipter probe or reporter, which I don't have time to explain, but believe me, we can correlate the fluorescence lifetime of this molecule with membrane packing and lipid order. So I will be showing you mixtures of POPC with both ether lipid or ceramide. And what we can see is that when we add ceramides to the liposomes, we see a decrease in the lifetime. And when we add ether lipids, this time at bigger concentration, because we understand the effect of ceramide in membranes is much stronger, we see changes in the same direction, confirming that indeed at least with this technique, we can see common effects on membranes of these two lipids. And so I think we can conclude that indeed sphingolipids and ether lipids seem to be co-regulated in order to maintain the homeostasis of a particular process, which is the secretion of GPI anchor proteins through the binding to P24 complex. And that this co-regulation can be seen also through evolution, where a ceramide molecule has been substituted by an ether lipid to perform the exact same function. And I believe, again, that nature is doing this because of their physicochemical properties and the effects they exert on membranes. And with this, I just want to thank everybody in the Risman Lab, current members and also past members that are already gone, and they helped me a lot during this time. And I want to thank everyone involved in this project, especially Jonathan and Manuel for starting this with us. And I want to thank the funding and of course you for your attention, and I'm happy to take questions.
All right. Thank you a lot, Noemi. It's been a great and clear talk. So for the participants, please type down your questions and do raise your hand for us to be able to unmute you. So it sounds like we've got questions from Christiana Andriani.
Oh, I think I was unmuted. I don't know if y'all can hear me.
Okay. Yeah.
So yeah, fantastic talk. You have a lot of really nice data, especially the combination of lipidomics and genomics and all of that. One thing that was a little bit confusing to me was in a lot of your screens, you identified ether PCs as very interesting, and you did a lot of great work in the biophysics and synthetic membranes. What is the link between the ether PCs and the GPIs, which my understanding were ether PIs? Do they interact? Are you using ether PCs as a model for ether PIs?
Not really, and I know that it's an ether PI. I just I don't think we know which are the exact precursors for the synthesis, but definitely we know that when you inhibit ether lipid synthesis in general, the GPIs don't get remodeled to have an ether anchor. And I think that these are two different things, okay? I think what I'm seeing is indeed ether PC or other ether lipids as well. I guess it depends on the cell types, for example, the particular ether lipids that are expressed. But what I see is that indeed, I see the effect of free ether lipids in the membrane surrounding the transmembrane domain of P24. So this is independent, but I think this is going to be an important factor to recruit GPI anchor proteins that have an ether lipid anchor, regardless of the precursor that has been used by the cell for synthesizing these GPI anchor proteins.
Gotcha, thank you.
So we've got a question from Jason Rodenkal. Jason?
Hey, this is Christine. I think Jason has already asked the question before.
Okay.
Noemi, terrific, brilliant presentation. I was just wondering whether did you see if any acyl chain composition and length of the other PC matter, and whether do you see a specific enrichment of certain other PC species after depletion of SMC18, or it's a generalized other PC enrichment?
So you mean if I see other lipids also upregulated upon myriosin treatment when I deplete the sphingolipids?
No, regarding the other PC. So do you see an enrichment as a other PC class, or do you see just specific species? So some length of the carbon backbone or unsaturation of the acyl chain, does it matter for your composition?
Yes. Yes, I think it does. And of course, we see many other lipids changing. It's just that we decided to focus for this project on ether lipids because we thought it was new. But of course, I see many other changes in other phospholipids. And one of the changes is actually the acyl chain length. And I think it's really well, how can I say, demonstrated also with the miriosine screen with the genetic data in the plot I showed you, with different telogases with different substrate specificity. I think this is showing us indeed how the cells cope with the lack of sphingolipids and how the cells, modify probably or remodel the phospholipids, the other glycerophospholipids in the membrane to maybe more saturated when we don't have sphingolipids. So yeah, that's something we see it's happening. Yeah.
And perhaps you should like look also into the peroxisome contribution, because other lipids are bed oxidizing to peroxisome, and especially if you say that, I mean, it's long chain other phospholipids, other lipids, it's very likely that peroxisome will play a role in your effect.
Sure, probably. Yeah, I agree. Thank you
so much.
I have a question regarding the biochemistry of the regulation. I find it fascinating that the cell can kind of feel that either sphingolipids or ether lipids are depleted. How do you think does this work? Is there a sensor for some biophysical properties of membranes or specific sensors for these lipid classes? What are your thoughts about this?
Yes, for sure. There must be sensors. There is some information out there, but overall, I think that's one of the big issues and long-standing questions in membrane biology and lipid biology. There are sensors for sure, and we need to start identifying more. But there must be a sensing machinery to rearrange the lipid composition of membranes. Yeah.
Okay,
whether it's the biophysical properties or also there is signaling involved, which is probably the case, that's something that we should take a look.
All right. So seems like we've got many other questions here. So we're gonna take like 2 more questions for the sake of time, and you can answer for the other questions on the chat if you want.
I don't see them.
Okay.
The questions.
So you have a question from, let's say, Tasanka, please. Are you hearing me? Okay. She's not unmuted yet. So Gregor?
You didn't hear me, okay.
Yeah. Yeah. Here you are.
No,
Okay,
Go ahead.
Hi, this is Sasanka Ramnathan. I'm at UAB, and it's a fascinating talk. I'm sorry, but I missed the front part of it because I couldn't log on. My question is, if you may have covered this already, so I'm wondering if you can use your whole setup to look at the mobilization or translocation of these sphingolipids in, let's say, in a stimulated cells that are undergoing stress. And would you be able to differentiate between, let's say, de novo pathway versus sphingomyelin hydrolysis or the salvage pathways as the pathways that generate ceramide?
Yeah, that's something that has to be done. We so far don't know. We have been depleting sphingolipids with miriosine so far, so of course that's something I don't know. Regarding the sphingomyelinases, actually I did some experiments that I didn't show you today, but that prove that indeed the decrease in sphingomyelin 18 we see when we deplete ether lipids might be being degraded in the lysosomes, because when I knock down the acid sphingomyelinase, we recover that sphingomyelin. But yeah, we didn't look much further. I don't know if I answered your question.
The reason I think it's interesting is that there are different disease disorders that that involve like the de novo pathway versus the sphingomyelin hydrolysis pathway. I think you have a very nice system to kind of
Yeah,
solve that.
yeah.
Thank you.
Good.
Agreed.
Thank you.
So next question from Bings.
Oh, hi. Yeah, that was a fantastic talk. I'm wondering whether the, with the ether lipids driving P24 one way and sphingomyelin driving it the other, whether that actually is important for the recycling. Does that drive the recycling of P24? So I guess in that sense, if you accumulated ether lipids in the Golgi, would that prevent recycling of P24?
You mean if we check the localization of P24 under those conditions?
Yeah, or just the general concept of having the ether lipids drive P24 in one direction and sphingomyelin driving in the other, that actually is critical for the recycling pathway.
That's one possibility. The problem is that it's a bit difficult to disentangle the retrograde transport from the anterograde, right? Because you will be affecting both. Most of the times when one is wrong, the other one can be affected too. That's one of the problems. But I picture the role of ether lipids indeed helping the anterograde transport of GPI and coproteins being more specific, perhaps from certain regions in the ER that are enriched on ether lipids and as well as GPI and coproteins. About the recycling, I'm not so sure actually.
Thank you.
All right, great. So I think I'm gonna stop there for the questions. Sorry about that. So Noemi, I think you can just type down your answers into the chat. Thanks again for the great talk. It was really very interesting.
Thank you. Okay, stop
So,
sharing.
yeah. Next speaker is Yilong Zhu. So Yilong is just like about to start his lab in China. So Yilong has been trained in China, so he went to New York for his graduate school at Pettering School, working with John Masage, with whom he's done his PhD, and then he went for his postdoc at Harvard, working with Stuart Schreiber, and he's like a multitasking guy. He's been working on cancer biology, metastasis, some structural biology and so on and so forth. And he has a huge interest on ferroptosis. So we are looking forward to hearing you, Yilong. Please, the stage is yours. Yilong? Yeah, we're not hearing you. You need to unmute yourself. Not yet. Not yet. Well, we are still not. Yeah. All right.
Can you hear me now?
Yeah, perfect.
Yes.
Yeah, cool. Thank you. Sorry for the technical glitch. Let me, and can you see my slides?
Yeah, perfect.
Okay, great. Thank you. So thanks for the nice introduction and for the invitation from the organizers. It's actually 2 a.m. In China now, so I apologize that I may be responding slower than usual. But if you caught any part of the presentation that's not clear, please feel free to ask me anything during the question session. So it's my pleasure to share with you our recent work on exploring the function and the plasticity of the polyunsaturated lipidome in cancer. So all our cells in the body are surrounded by a membrane that is mainly made of lipid molecules. And the similar membranes are also surrounding many of the organelles inside of the cells. So it's certainly important for life that these membranes are impermeable to external molecules, but membranes also need to be flexible and fluidic enough to permit cell division and membrane fission and fusion. So these features are largely achieved by introducing unsaturated fatty acids to the membranes. According to the number of double bonds, fatty acids can be grouped into saturated, monounsaturated, or polyunsaturated fatty acids. I'm going to use MUFA or PUFA as an abbreviation for these terms. And as the unsaturation level increases, these fatty acids display decreasing melting temperature and the packing density, as well as increased curvature and fluidity. At the cellular level, the introduction of polyunsaturated fatty acids or PUFA into cellular membranes is very significant in evolution because it's very effective in enabling motility, vesicle fission and fusion, as well as the formation of bioactive lipid signaling molecules. However, there's one major peril of inducing polyunsaturated or PUFA lipids in membranes, which is that the multiple double bonds organized in these pentadienyl structures can make PUFA lipids particularly susceptible to hydrogen abstraction and lipid peroxidation. So one reason for lipid peroxidation to be particularly concerning is that in the presence of oxygen and reactive iron, lipid peroxide radicals can self-propagate and lead to a burst in lipid radical species, which damages the cellular membranes and proteins and causes a cell death process called ferroptosis. So at the organism level, lipid peroxidation can contribute to various degenerative diseases in the kidney, in the liver, and in the brain, as well as to the normal aging process. So in order to balance the ferroptotic risk, eukaryotic cells have evolved a counter enzyme called glutathione peroxidase 4 or GPX4 in order to reduce lipid hydroperoxides to non-toxic lipid alcohols and keep cells alive. Therefore, artificial inhibition of GPX4 can effectively induce lipid peroxidation accumulation and ferroptosis. So here is just a brief video showing the morphological processes that how a cancer cell can undergo ferroptosis after exposure to a GPX4 inhibitor. It certainly highlights the massive membrane changes that was happening when the cells are exposed to GPX4 inhibition. So our cells really need to keep a fine balance when utilizing PUFA lipids in order to avoid aberrant peroxidation and ferroptosis. So this balance is much more vulnerable in cancer cells than in normal cells, largely due to the increased usage of unsaturated lipids in cancer cells. I'm just showing one example here in a mouse non-adenocarcinoma model, where tumors induced by KRAS activation can exhibit significantly higher levels of PUFA lipids, including DHA and arachidonic acids, as well as lipids that contains these two different fatty acids compared to normal lung tissue. This is shown by a mass spec-based imaging technique. So increased unsaturated lipids are also seen in cancers from the breast, ovary, and etc. These collections that prompted us to ask, can we leverage the high PUFA lipid state to target the cancer via ferroptosis? So at first, can we actually identify a group of cancer that is intrinsically sensitive to ferroptosis? We resorted to Cancer Therapeutics Response Portal, which is a chemical biology database developed here at the Broad Institute. And this data set contains the sensitivity profile of 545 selective compounds with unique mechanisms of actions over 900 cancer cell lines. And this compound collection contains at least 3 GPX4 inhibitors listed here, including ML210, and then several other ferroptosis-inducing agents. So using simple comparative analysis by looking for ferroptosis sensitivity across different lineages, we found that cancers from the kidney, the ovary are among the most sensitive to ferroptosis. And the top sensitivity occurs in a group of cancers called clear cell carcinomas, which are featured by the presence of abundant lipid droplets in the cytoplasm. So in fact, the clear cell morphology is defined by the blank cytoplasmic space resulted from lipid depletion by organic solvents during histological analysis. So clear cell carcinomas most often occur in the kidney, in the ovary, and also occasionally in the other tissues such as pancreas, liver, or thyroid. And together they kill about 15,000 patients annually in the US. The therapeutic challenge for managing clear cell carcinomas include that they often present as metastatic disease, and particularly in the context of kidney cancers, they develop resistance to therapies very rapidly. So we, based on our bioinformatics finding, we extensively validated the dependency on GPX4 in clear cell cancers in vitro and in vivo. For example, in contrast to the wild type 7860 clear cell renal cell carcinoma model, GPX4 knockout 7860 cells were not able to colonize the mice and develop tumor. However, this defect can be rescued by treatment with lipiroxystatin 1, which is a lipid radical scavenger and a frequently used ferroptosis inhibitor. And when we withdraw LIP1 from the mouse treatment, the tumors once again can regress and through ferroptosis, suggesting that GPX4 might be a therapeutic target in clear cell cancers. We're quite interested in what makes clear cell cancers particularly sensitive to ferroptosis. So with similar approach as Naomi has mentioned, we performed a series of genome-wide CRISPR screens in both kidney and ovarian cancer cells. And there are apparently two promising gene modules that showed up from the screens, including HIF2 alpha module and the peroxisome module. We also found several other genes that appeared to be interesting for ferroptosis modulation, and I will briefly touch on that. But before I dive into these pathways, I just want to give a quick shout out on a new computational method that we developed along the way while analyzing this CRISPR data. So this method is called gene list network enrichment analysis, or GenLinear in short, as we call it. What GenLinear allows us to do is to nominate gene signatures and pathways from a list of genes that emerge from an RNA-seq or genetic screening experiment. So this is very much like what we have been doing for a long time called gene set enrichment analysis, or GSEA. And GenLinear is significantly more powerful in our hands compared to GSEA, because it not only counts the identity of the genes that were overlapped between the gene list and a target gene set, but it also takes account into the protein-protein interaction information, both physical interaction and functional interaction, which is resulted from databases curated by, such as String. So we have recently released the code of GenLinear on GitHub. So please feel free to try out these computational codes. And we're also in the middle of developing a graphical user interface for non-programmers to use. Coming back to the pathways identified by the CRISPR screening. So the HIF2 alpha pathway first got our attention, and it's particularly interesting because the HIF2 alpha pathway has been known as the oncogenic driver, at least in clear cell renal cell carcinoma, which is often caused by VHL silencing or mutation. And when we knock out HIF2 alpha, we found decreased sensitivity to ferroptosis, which is a validation to the CRISPR screening. And more interestingly, HIF2 alpha knockout can somehow selectively downregulate the polyunsaturated lipid proportion, but not really the saturated or monounsaturated fraction in these cells using lipidomics analysis. And this difference is more prominent when we look at the free fatty acid level, as shown here. Free PUFA, but not free saturated or monounsaturated fatty acids are downregulated when are dependent on HIF2 alpha. So we also mapped the downstream genes, the downstream target genes of HIF2 alpha, which is responsible for this PUFA selective regulation, to be HIO-PDA, or hypoxia-induced lipid droplet associated protein. A small fragment. This HIO-PDA is a small peptide that's very that has been very unknown in terms of its biochemical functions. So we are kind of in the process of exploring what are the exact biochemical mechanisms for HIO-PDA to act in this way. Altogether, these findings suggest that although HIF2 alpha is driving tumorigenesis in kidney cancers, one of its lipid metabolic traits is to lead to a targetable vulnerability for the induction of ferroptosis. With regard to the second pathway, which is the peroxisome pathway, we found that genes including encoding peroxisome components are significantly enriched, including structural components such as PAX genes, many of the PAX genes, as well as enzymes such as AGPS, FAR1. You are familiar with AGPS from Naomi's presentation already. And also genes that are peripherally associated with peroxisome, such as ACSL4, which is also a well-known modulator of ferroptosis and the polyunsaturated lipid regulatory enzyme. And indeed, when we disrupt peroxisome biogenesis by knockout PAX3 or PAX10 or many of the other genes that we validated, the cancer cells become more resistant to ferroptosis. So notably similar effects were also seen in, we tested a few other cancer models, including liver and endometrial cancer cells. Peroxisomes are particularly interesting because they are multifunctional organelles involved in many aspects of cellular metabolism, including lipid synthesis and degradation, as well as RS detoxification. Then how does peroxisomes contribute to ferroptosis? So we went back to our CRISPR screening and found that nearly every component of the either lipid or the plasmalogen biosynthesis pathway, including FAR1, GNPAT, and AGPS, are very high on our list, suggesting that peroxisomes could be contributing to ferroptosis by synthesizing either lipids or plasmalogens. And thanks to Naomi, who has very nicely introduced the differences between either lipids and ester lipids, I just want to point out that besides the differences of using one alkylyl ether link for plasmalogens, rather than using the ester link in diacetyl phospholipids, the plasmalogens often have polyunsaturated lipids at the SN2 position. So this presumably making these lipids susceptible to lipid peroxidation and ferroptosis. Indeed, when we knock out plasmalogen biosynthesis genes such as AGPS, we can recapitulate the lipidomic phenotype as well as the ferroptosis resistance phenotype conferred by peroxisome disruption, suggesting that the effect of peroxisome in ferroptosis might be largely regulated by its role in plasmalogen or either lipid biosynthesis in general. So the involvement of plasmalogens in clear cell carcinoma ferroptosis may also have clinical relevance, because when we reanalyzed a previously reported lipidomics data set of 49 matched pairs of clear cell renal cell carcinoma tumor versus normal patient samples, we found that plasmalogens or either lipids in general are among the most upregulated phospholipids in the tumor samples. In addition to the HIF2 alpha and the peroxisome pathways, we also found several other genes. I'm not going into too much detail, but we do find one of the key enzymes called cytochrome P450 oxidoreductase appears to be essential for ferroptosis occurrence. And when we knock out POR, the cells are no longer much more resistant to ferroptosis. So we later mapped this process to be that POR might be directly contributing to the lipid peroxidation process. We are still not sure whether it requires the classical partners of POR, such as the CYP, the cytochrome P450 genes, the proteins, but this is a process of high interest to us. And the benefit of this finding is that it does offer a positive regulator for ferroptosis, so we can better design therapeutics for inhibiting ferroptosis in our cells. So the next question that we're really interested is, given our findings that clear cell carcinomas are highly dependent on GPX4, and we are in the process of developing bioavailable GPX4 inhibitors, we wonder how durable are the effects of GPX4 inhibition in restricting tumor growth. In order to explore this process, we created a GPX4 knockout cells because we don't really have a bioavailable GPX4 inhibitor for in vivo use. We used these GPX4 knockout cells to anticipate and implanted these cells to the mice, anticipating that the cells would undergo ferroptosis. And indeed, initially, we saw no tumor growth in the beginning. However, when we wait for long enough, in this case, it's about 8 to 12 weeks, all the tumors actually grew back and they eventually killed the water mice, suggesting that cancer cells initially sensitive to ferroptosis can evade cell death in vivo. And importantly, when we re-isolated the cells from the resistant tumor, the relapsed tumor, and re-implanted back these cells to the mice, the tumors grow rapidly even without latency, suggesting that the cells have acquired certain inheritable traits that are rendering them insensitive to GPX4 depletion. We're pretty sure that these cells are still GPX4 independent because by Western blot, the GPX4 protein remains null even in the second round of isolation. So what are such traits that makes these cells insensitive to GPX4 depletion? We performed many omics analysis, including exome sequencing, metabolomics, and gene expression analysis. And using lipidomics, we found that the ferroptosis-resistant cells have significantly downregulated their plasmalogen levels, shown here in blue, and the ones, particularly the ones that contain polyunsaturated fatty acid chains. This is occurring without significant mutations using our exome sequencing analysis. So this finding is coincidental with our initial findings through CRISPR screening, showing that the plasmalogen and the peroxisome pathways can regulate ferroptosis sensitivity in the same cancer cells. But how are plasmalogens downregulated in these ferroptosis-resistant cells? So we then performed RNA-seq analysis and found that at least several either lipid biosynthesis genes are downregulated in the resistant cells, including TMEM189, which is a recently identified phosphatidylethylamine desaturase involved in actually creating the alkylyl ether bond in ether lipids, as well as other genes such as AGPS and ABCD1, which are all involved in different segments of the ether lipid biosynthesis pathway. So importantly, when we co-deplete GPX4 and any of the ether lipid biosynthesis genes, we found that the double knockout tumors can grow much faster in vivo than the GPX4 single knockout clones, indicating that the downregulating plasmalogens is an effective way to evade ferroptosis in vivo. This result suggests that the originally ferroptosis-sensitive cancer cells can become ferroptosis resistant by downregulating their plasmalogen levels. And at this point, we do not know whether this mechanism of dynamic plasmalogen regulation can also happen in patients that are exposed to ferroptosis inducers. So the plasticity we kind of find that the cancer cells are somehow exhibiting a certain level of plasticity in their plasmalogen levels, as well as the sensitivity to ferroptosis. So we are intrigued by this finding, and we wondered whether there are other contexts that plasmalogens can also regulate ferroptosis sensitivity. It was previously suggested that neurons and cardiomyocytes can exhibit the highest level of plasmalogens relative to other cell types in a human body. So we then explored these cell types using in vitro differentiation systems. And during neuronal differentiation, we found that differentiated neurons are much more sensitive to ferroptosis than their undifferentiated progenitors. And this process is interestingly coupled with significant upregulation of plasmalogens or polyunsaturated ether lipids in general, ether phospholipids in general, during this process. And a similar trend were found during cardiomyocyte differentiation, where we used iPS cells to induce them to differentiate into cardiac progenitors versus a maturely differentiated cardiomyocytes. So this is kind of interesting, but we don't quite have a good idea about how exactly these plasmalogens or polyunsaturated ether lipids would contribute to the physiology of neurons and cardiomyocytes. To this point, my research has highlighted that PUFA lipids play important roles in driving ferroptosis sensitivity and shaping cell states. But how can we rapidly detect cells that contain high levels of polyunsaturated lipids? Since single-cell lipidomics is still at its infancy, we developed a technique called photochemical activation of lipid peroxidation, or PULP in short, to enable the rapid detection of PUFA lipids. So this work is done in collaboration with Dr. Liu's group from Tsinghua University in Beijing. And the key innovation in PULP is that we can use focused high-power laser to induce lipid peroxidation photochemically and non-enzymatically, which will be captured by a fluorescent reportable lipid C11. And this way, the lipid peroxidation signal we get will only be proportional to the local PUFA lipid concentration, but not on the expression of the enzymes, which the expression levels may vary between different cells. So as shown in this video, after 5 seconds of nasal illumination, it induces a strong lipid peroxidation signal, which is white here, indicated by this this spot. And also, as a proof of principle, by knocking out ACSL4, which is a genetic way of depleting polyunsaturated, at least esterified polyunsaturated fatty acids in the cells, we found that PELP can only result in a signal in a high PUFA cell, but not in a low PUFA cell. So we envision that this technique can be useful in rapidly detecting and reporting polyunsaturated lipid levels in heterogeneous cell population, and also to study ferroptosis mechanisms at high spatial temporal resolution. And we recently are able to validate this technique to several more cell lines that are exhibiting high levels of PUFA. So with that, I would like to thank many of my collaborators. I want to give a special shout out to Paul Clemens and Vlado Dancic, who are computational chemists that designed the Geneva computational algorithms. And I would like to thank Clara Clich and Amy Dick, who helped us to perform the metabolomics analysis, and especially Bob Weinberg and Whitley Henry, who helped us with the peroxisome and plasmalogen study in ovarian cancer. I would like to thank my funding source as well, and this will be the information for my new lab, which I will start next week. So thank you very much, and I'm very happy to take any questions.
Right. Thank you a lot, Yilong. Well, again, if you have questions, please type them down in the chat box and raise your hand. So sounds like we've got a question from Marcos.
Yeah, let me see.
Please, Marcos. Okay.
Marcos, you should be unmuted.
Well.
Hello?
Yeah.
Hello? Go ahead, Marcos.
It's not unmuting. Oh, hello. Okay. So nice talk. I actually have been following your work a little bit. I had a question in the context of TMEM 189. When I read your paper, maybe you can remind me, but I don't think, I wanted to see, and maybe you tried this, but have you tried doing a functional study where you knocked it down or knocked it out and then tried seeing how that modulated the activity? So for example, in my question, I said that it acts as an antioxidant as well, and you're postulating that it's potentially acting much like a PUFA, where you're essentially oxidizing that vinyl ether bond. And if you've tried incorporating exogenous PUFAs to see if they're more sensitized with that knockdown, or exogenous MUFAs to see if they're potentially protected, because you probably know that the Dixon lab found that MUFAs were involved in a protective function, specifically in the plasma membrane, is their final hypothesis from that study.
Yeah, it's a very interesting question. So first, answering your question about TMEM 189. So we were quite intrigued by TMEM 189, given its recent findings at a desaturase, which actually results in the double bond at the SN1 position. So first, it's not emerging from our CRISPR screening, but we still pulled it out and then knock it out using CRISPR shRNA. We also tried to rescue it. We really didn't find any significant effects on ferroptosis sensitivity, suggesting that the double bond at the SN1 position may not be really contributing to ferroptosis sensitivity at the same scale as the polyunsaturated proportion. And it's understandable because usually the ability to be peroxidized goes up exponentially when you have multiple double bonds that are organized in a pentadienol structure. But the SN1 double bond is basically a singular position. We also did a few other functional assays. Used by the most important one is by delivering full phospholipids, which include diacyl phospholipids, isophospholipids, or alkylyl isophospholipids into the cells. We did not find the presence or absence of the double bond at the SN1 position, meaning the alkyl ether versus an alkylyl ether. We did not find that to be affecting the ferroptosis sensitivity. Instead, the SN2 position fatty acid chains appears to be the dominating effect. So this is certainly, I think it does contradict slightly with the existing concept that the SN1 position alkylyl structure might be acting as like cell membrane intrinsic antioxidants during oxidative stress. But I think it's probably now a more quantitative difference, meaning that when the cells are not under ferroptotic stress, like they have a kind of a basal level of oxidative stress, the SN1 position might be sufficient to act somewhat protectively to oxidative stress. But ferroptotic stress is a very strong and prominent, and also it goes up exponentially. So that level of oxidative stress may be too strong, so strong that it covers the effects from the SN1 position, if I'm making myself clear.
Yeah, so you're saying that it's a homeostatic modulation, essentially, where if you have too much, and I think I've read that in a review somewhere, is we're trying to work with some TMEM mutants on our end in another model organism. So I was really curious to see what you guys found as well. So yeah.
Oh, sure. Yeah. I'm happy to discuss more on that end.
Yeah, I'll probably email you later with questions.
Sure.
Okay, sounds like we have a question from Clarissa.
Sure.
Clarissa Mayer Montero.
Mute. So can you hear me?
Yeah.
Perfect.
Okay. So it's very, very interesting, this pulp technique. And have you done, two short questions, one, and have you done it with the clear cell carcinoma cells? And could it be useful to distinguish between heterogeneity in the tumors? Because we know that tumors are not one cell, or at least not the same cell in the same stage of development. So then it would be interesting to check for the heterogeneity.
Yeah. So let me answer this question first. We are very excited about this technique, but due to the pandemic, we haven't moved much far from cell culture. So we are using basically single layer cell culture. We have not tested it at a tissue level, but at a cell level, we are able to look at heterogeneity in both two levels. One, subcellular level. So if you have a confocal microscope that you can split your laser into, let's say, 20 different spots. I know many confocal microscopes can do that. You can split it into different spots in the cell, so that you will be able to see different signal levels at a different location. And secondly, if your cells are on the same plate, you will be able to look at them, different cells across a few different, like you can look at across different cells. The challenging with looking at the different cells is that because we know that if you hit at a different subcellular structure of a cell, you get different a different signal. So when you look at different cells, it's hard to sort of normalize it.
Yeah. Okay.
Please.
And then the lipids within the lipid droplets, because I work with lipid droplets, and we have a work with cancer cells, lipid droplets, and you have also heterogeneity between the lipid droplets. Could it be that some lipid droplets are protected from this, especially the smaller lipid droplets could be protected even with lots of polyunsaturated fatty acids? They are protected to oxidative stress.
Yeah, this is a very good question. It's a very good question too, and I think it's being explored by many different labs in terms of the relationship between like how active are the lipids in lipid droplets during these processes. So that's a kind of a common question. In our hands, it's not reviewed by pulp, but we have several other data that we seem to lead to that lipids that are stored in lipid droplet, particularly the polyunsaturated fraction, does not appear to directly contribute to lipid peroxidation. It more seems to be the lipids that are in the ER under the plasma membrane. We don't quite know whether, for example, mitochondrial lipids would be directly involved yet, but the ER appears to be definitely necessary and appears to be initiating this process when the cells are stressed. And a second comment on the using whether pulp can be used for lipid droplet. So the technique is relatively easy, but we don't have resolution at the Z scale, meaning that when you hit a cell, the laser goes through the bottom plasma membrane all the way through the top plasma membrane. So we can't quite tell at which Z level it's actually acting. Yeah.
Interesting. Thank you very much.
Sure.
Yeah, great. Yeah, sounds like you've answered all questions. I mean,
oh, cool.
Yeah, I have one question. I mean,
sure.
Have you ever like tried to following up on this question to like boost or prevent lipid droplet formation and see how that impacts? I mean, the cell death basically.
To improve lipidome in order to affect cell death, you mean?
Yeah.
Yeah, that's an interesting question. So we do frequently use, for example, supplementary polyunsaturated fatty acids or even phospholipids to sort of titrate the sensitivity to ferroptosis. That has been very effective. So I think we have seen that polyunsaturated fatty acids are able to increase the sensitivity, and then Marcos has mentioned that monounsaturated fatty acids are very effective in preventing ferroptosis. So we seem to be able to achieve semi-quantitative titration of that sensitivity in cells, not so much in tissues yet.
Great. So I guess we're gonna thank you again. And
may I ask one more question on behalf of someone?
Oh, yes.
Yeah, this is for Jacob Cook asking this question. In the tumor samples, were you able to distinguish between plasmanal and plasminal lipids in the highlighted ether lipid species?
Yeah, that's a good question. So we have not gone that far. We are quite interested, although that in those tissue, just based on the literature, it appears that plasmanal, meaning the alkylyl ether lipids, are often the most abundant ether lipids in human cells, especially when they come to polyunsaturated proportion.
Oh.
Yeah.
Thank you.
Thank you again, Yilong. And
okay, thank you, Rachel, and thank you, the organizer,
for your installation. And we thank all the attendees. And see you in 2 weeks potentially.